So thank you, Ele, for the nice introduction. Next slide, please. So just briefly, before going into the programs and the application of AI in 4 different examples of drug discovery in oncology, some few words about Iktos and the technology and the platform that we have developed for improving the delivery of new drugs and new molecules in drug discovery. Iktos is a French-based company that has been created in 2017, nearly 8 years ago. We are approximately 50 people with a lot of very strong focus on AI and data science people, drug discovery expertise, medicinal chemistry, and chem team, of course, plus a big group of developers. The core of the business of Iktos is really to develop AI technologies and automation in the labs to really help the drug discovery process to be improved and to go faster to the hit to lead and lead optimization using the AI technology. So we have two main businesses, which are the drug discovery collaboration and, of course, also our own pipeline that is open for licensing, and the development of the application of the software and the AI solution we design within Iktos to make it available through SaaS and licensing. We have some groups, of course, most of the people are based in France, and we have also a small team in Japan and a small team in the US. Next. So what is Iktos about? It's at Iktos, we have been really keen at developing cutting-edge AI technologies to help the drug discovery processes at many stages. The core of the technologies is, of course, the generative AI, which is embedded into a software so-called MACHIA. And very soon at Iktos, there has been the consideration that AI without chemistry knowledge and chemistry awareness by the AI of the synthesis and the synthetic issues could be a kind of blocking part of the process. And so this is why we also developed a retrosynthetic analysis tool, which is so-called SPIA. Lately, we also developed a robotic and orchestration platform AI that help to execute the synthesis of the molecules that are generated by MACHIA. So overall, we have developed a completely comprehensive platform that goes from the generation to the retrosynthetic analysis to the synthesis through an automation platform. We are currently developing the automation, also the purification, and recently we have linked to the process a kind of new technology for assessing the protein-protein interaction and induced proximity within the cell to help testing the molecule and generate more data to feed the generative AI. So this is basically a very short view of our current platform. Next. So how does it work in the principle of the generative AI? The very important point about the generative AI at Iktos, that we have really considered the synthetic accessibility of the molecule that can be generated through the algorithm as a core component of the system. So basically, you will see with an example, in the past, we developed, as many other companies, LLM and LSTM models that was more or less based on a kind of footprint, SMILES footprint of molecules. And we have really developed the algorithm through neural networks to something that embed the chemical knowledge of a molecule within the generation. And so today, we have a generator that knows how to make molecules, and the molecules are generated based on this knowledge, which means that basically the molecules that are generated under some specific reward are by definition synthesizable, and it's the result of the combination of 3,600 organic reaction and the potential one commercial building blocks available to make the synthesis of these different combinations. So there are virtual reaction, virtual building blocks associated through a specific target product profile. We want a molecule X that binds the protein X with a molecular feature that can be medchem-like, that we embed also in the generation. And of course, we score and we reward the generation through a lot of different systems that are not just docking, but we have also associated some reward functions that are AI-based, such as folding, co-folding. Of course, we can use the BOL system, but we have also developed our own contact score and Iktos 3D so-called denoiser. And so we make the combination of all those scores to focus the generation on the most potentially active molecules. We can also, of course, help take the benefit of pharmacophoric score, similarity, alignment, and so on. So we have a lot of different features that can be incorporated into the generation, always driven by the synthetic accessibility of the molecules to deliver the optimized molecule. So now we are going to look at so the way we use all those tools in the context of the robotic platform. Next. So basically, the point was that we realized that somehow a kind of automate, or let's say an automatic systems could be able to synthesize the molecules once they are generated. And so this is what we did. The concept is that a molecule generated under the specific score and reward function that we described in the previous slide can be retrosynthetically analyzed by SPIA. And then another algorithm, so-called ILACA, can order the molecules and organize the synthesis using a ChemSpeed robot to execute the synthesis of the molecules. And here, this is not just parallel chemistry, because what ILACA can do is really to understand, for instance, to make the synthesis of 96 different molecules that will have just one common feature that can be, for instance, the temperature. So you can carry out different syntheses with completely different pattern in terms of reactivity using the same plate. This is the principle of ILACA. So then we will go through some example of the how we use the AI combined with the medchem expertise in the context of 4 different programs where there have been some classical synthesis and robotic execution of the synthesis to deliver new hits or new lead. So we are going to cover 4 examples of hit disco or lead optimization with some different target, 2 kinases, PIM and PKMYT1, Polymerase Theta, and PDL1 small molecule binders for immuno-oncology. Next. So PIM kinase. Next. So again, this is really a case study. The PIM kinase is potentially a very interesting target, but nevertheless, the experience in the past for many therapeutic programs driven by carried out by many pharmas was not really conclusive, and we will probably explain why. But this is here really an example of the demonstration of a specific feature that was create novelty within a crowded space. So what are the PIM kinases? This is a family of 3 isoforms, PIM1, PIM2, PIM3. They are non-essential serine kinases with a lot of biological pathways more or less controlled by the PIM kinases. The PIM kinases can be activated by many different factors, growth factors, cytokines, mitogens, and hormones. And as you can see on the cartoon on the right, there are a lot of pathways where PIM kinases have been shown to be involved, sometimes critically, sometimes not. And the most of the association has been linked to oncogenic pathways. There have been the demonstration that PIM1, PIM2, or sometimes PIM3 can have a pivotal role in proliferation in cancer cells, metastasis, also chemoresistance. More recently, there have been also the report of some potential involvement on immuno-oncology or immunoinflammation. So again, potentially a broad application, but a lot of questions about the real criticism of the target. So for sure, PIM overexpression is really associated with several cancers and hematologic malignancies. So this is why there have been a lot of projects. But today, after several clinical trials by some companies, the efficacy has been shown to be limited, especially because of the tolerability and also potentially the clinical effect. Some of the points addressed by the competition was really about the lack of selectivity over the different isoforms. And this is why there are still some activities about the research of potentially more selective and safer isoform inhibitors of PIM kinase. So next. So our goal was really to say, okay, here the exercise would be to help, thanks to the AI, to use the generation of molecules that would be truly novel after a careful analysis of the chemical space covered by the molecule described in the competition. So there are several chemotypes, as you can see. This is just an exemplification with some examples. There are far more than this. Most of the time, the molecules are not selective across the three isoforms. There have been a lot of molecule progress into the clinic, and as you can see, big players such as AstraZeneca, Novartis, Amgen, Genentech, and so on have been working for a while in that area. And the goal was really to say how we can find out using AI a completely novel scaffold within this very crowded space using the 3D GenAI. And of course, once we have selected or at least identified the scaffold, see whether or not to try to understand why they would be or they could be selected. Next. So what we did, so at this time, it was the very last example for which we used a generator that was not yet the one we are using now based on the chemical reaction. Here we trained a model of generative AI that was made using an LSTM generator trained on Campbell, in which we also incorporated a very strong enrichment of PIM kinase inhibitors. Basically, we injected 3,600 data points of molecules that were described as PIM inhibitors, as a kind of, let's say, superpopulation of, let's say, chemical knowledge of kinases and PIM inhibitors in addition to the Campbell database. Of course, we also used reward functions that are medchem-like. This is something that we do for all the programs. We try to make some rewards for the molecule that are the more in agreement with the medchem rules. Low molecular weight, good chromolog D or CELOC D, good TPSA, number of aromatics, and so on and so on. And we used for the generation in 3D specific docking score and custom Iktos so-called contact score. This is a system that we use to select residues for which we can put a specific weight on the specific type of interactions that can be, of course, salt bridge, edge bonding, cation P and P hydrophobic interaction, and so on. So here we made the analysis of the pharmacophore of two structures coming out from the PDB, one structure which was a Genentech inhibitor, and the other one an Amgen one, for which we can see on the table there were different features in terms of the type of residues that were targeted by the molecule, and we use this as the first generation to select the molecule to be synthesized. Next. So here we made two short generations. and we identified 4 different scaffolds, and we select 9 molecules for the synthesis. You can see the molecules are extremely not just simple, but they are really medchem-like, easy to synthesize, and this is why they were selected according also, of course, with their specific score. Hopefully, as you can see, we tested the molecule for just 2 concentration % inhibition on the 3 isoforms. As you can see here, that across the 4 different scaffolds, we quickly identified at least 1 family of this pyrimidine, this aryl pyrimidine, that were a bit displayed good activity. Across the 4 different scaffolds, we did not observe any activity of PIM2, and I will come back afterwards to this point. Nevertheless, we found 2, at least 3 different scaffolds with pretty decent activities as the first shot. So what we did at this stage was not to embark into the further expansion of one of the specific scaffold, but we used the knowledge extracted from this first result to retune the contact score and the different way to put on specific residues to make a third generation according to the first result. Next. So here, after making the analysis of the scores and the activity that was shown with this 3 scaffold, we remake a second analysis of the key residue to select. And again, we put a specific attention and increase the weight of the reward to some specific residues, sometimes hydrophobic, sometimes also such as the glutamic acid, with more weight into the salt bridge and potential interaction through hydrogen bonding. So we put a very high weight to the specific residues to carry out a third generation using the same model. Next. And so here, luckily, we nearly ended up with 1 scaffold, 1 privileged scaffold, this pyrrolopyrazole, which really emerged as the most privileged across the generation. And as you can see, with just 7 compounds synthesized this time, we nearly observed the beginning of SAR, and with a very strong reward given by the introduction of this amino piperidine on the right-hand side, which basically drives the potency, very likely through the interaction with a glutamic acid. So here, as you can see, we started with molecules that were at the micromolar range, and the most active one, Iktos 165, was pretty selective over PIM3 with a level of activity around the 10 nanomolar. So it was really extremely satisfying to see that with very, very few molecules synthesized, we were really able to achieve the goal. Next. So this is just to give you a bit of understanding of how we rationalize the activity. For sure, we really, the docking model and the contact score model really achieve a very nice interaction with the key residue selected, and especially the glutamic acid that was targeted by the molecule initially. Next. So overall, when we profile the molecule, we had also a pretty good result in terms of Chromolog-D solubility, pretty reasonable clean as a first shot, and also really a good selectivity score. We did a kinome scan at 10 micromolar, which is pretty high. So we assume that the selectivity would be even further improve at 1 micromolar or below. And also from a novelty standpoint, of course, the goal was 100% achieved. The scaffold is absolutely not represented in any patent or within the literature in the PIM area. Even more, in the kinase overall, we found very, very, very few examples of this scaffold in some patents and no application in onco or of course, PIM. So for many reasons, so we really consider that this project was very successful in terms of delivery. Next. So basically, the goal was to generate very high quality hit and developability in terms of potency and developability, and of course novelty. And here we were really hit all the targeted objectives in less than 20 molecules and less than 6 months with 4 different chemotypes. Next. So the other example is PKMYT1. So PKMYT1 is a kind of pretty hot target those days. It's a target that has been implied into in the context of CCNE1 amplified cancer. The amplification of the cyclin E1 is something that is observed in several gynecological and gastric cancers. Basically, what it does is that the amplification of the cyclin deregulate the cell cycle, which with a very strong and premature entry into S phase. So the cell cycle overall is deregulated, which is very often the case in many tumors. And the mirroring kinase is the WEE1, which is also a very hot target in the clinic. But the concept is that in the context of amplification of cyclin E1 cancers, the very specific phosphorylation of CDK1 for cyclin B by PKMYT1 could be probably more selective than the WEE1 that phosphorylate and that put a kind of brakes on all the cyclins with potentially more liabilities in terms of safety. So today, PKMYT1 has been considered as a very interesting potential target, which is synthetic lethal in the context of this cyclin E1 amplified cancer. There are several molecules in the clinic with lunresertib from Repair Therapeutics far ahead the competition. There is, of course, a lot of competition in that area. And again, we selected this target as also a potential way to demonstrate the value of using AI to differentiate in terms of scaffold or new selective lead in the context of heavy competition. Next. So here, this is to give an example of what was the challenge. Of course, there is lunresertib, so a very interesting molecule. And there have been a super strong competition with basically a lot of competitors that try to more or less capture the key pharmacophoric element, which include this exonitrogen, aromatic nitrogen, which is present nearly in all the competition scaffold, except from one, for instance, the azaindole. But most of the time, there is an NH2 present as a key pharmacophoric element in the molecules. So basically, what we did was to say, okay, how can we use our 3D generative AI to quickly identify and synthesize through the robotic platform molecules that would capture most of the key pharmacophoric elements while not having any NH2. Next. So this is a kind of summary. Of course, to create novelty, to avoid potential liability, which is frequently linked to aromatic nitrogen, and see how much we can also execute the synthesis of new molecules through the robotic platform and going fast. Next. So this is what we did. So the generation was, the foundation of the generation was the following. We identified the key pharmacophoric areas within immune receptor within the ATP site, but this is a very, very flat area, for which is very difficult. That would be extremely through aromatics, especially. So we selected some residues, and here, because we use the new generator, which is based on chemistry, we kept the phenolic counterpart, the part of the molecules, as a key element, and we decided to make the generation by growing through this specific chemical fragments with some reactivity that was allowed through, for instance, bromine or boronic acid, and by more or less by coupling or this type of chemistry. Next. So here, again, the first generation was pretty successful. We identified 14 different scaffolds through a set of 16 molecules synthesized and tested. We identified 5 molecules that were below the micromolar, belonging to 3 different scaffolds. And as you can see, one of the scaffolds, from EKT585, was around the 30 nanomolar range. So just one shot, 16 molecules synthesized through the robotic platform in 2 or 3 steps. And we basically ended up with a pretty decent outcome in terms of number of molecule active, and especially one very interesting series. So we decided then to use that first andazolopyrimidone scaffold as the source of the small heat expansion to see how much we can go into in terms of optimization. Next. So here, again, we used the understanding of the binding mode of lunresertib plus our molecules to really identify the key vectors and key areas for further improvement. As you can see very nicely, according to the hypothesis that we made, the NH of the ortho-orthoprime hydroxyanilines recapitulate a bit of the interaction of the NH2 of lunresertib. You can see also in the table that the first ADMET parameters of the molecules were pretty nice. Also the permeability CACO2 was not super high, but it was still, as a first shot, pretty decent. Even though also the clint was not that bad. So as a starting point, it was really a very good hit. So one of the key elements of the progression and expansion of the molecule was really to target again the 2 different carboxylic acid, especially an aspartic in the end of the pocket, for which we put a specific reward in that direction. Next. So this is again what was the result of this first round of expansion. As you can see on the left, growing through the carbon between the 2 nitrogen of the pyrimidone was not really a great outcome. No real big impact or jumping, or most of the time detrimental. So this is more or less according to the structure, not surprisingly. Around the phenol, no surprise here, we really capture the same type of SAR that from the competition, and repair, and very difficult to change the phenolic counterpart. There are some hints that might be something achieved with an andazole, but we did not went very far in that direction. And hopefully, through the expansion of the pyrazole part, we ended up with very nice result with two molecules that were far below the 10 nanomolar. We put 10 nanomolar because we are the limit of the IC50 quantification in the assay we got. Those two molecules make very nice interaction with the targeted aspartic, and so we were really happy to get those two very potent molecules. Next. This is just the docking and contact model analysis of the molecules, which is a very nice agreement with the potency. Next. So again, as a summary for this part, here we have a very nice demonstration of the strengths of using the AI and the execution for the robotic platform as a, let's say, first exploration of a potential new series. With less than 40 molecules synthesized, we were able to identify several scaffolds and one privileged scaffold with a single-digit nanomolar activities and a pretty good ADMET and physicochemical properties as a foundation of further E2 lead optimization. And now I will leave the floor to my colleague Christopher for exploring Pol Theta and PDL1 binders. Thank you.
Thank you, Alexis. So I'm going to cover Pol Theta project. So we already mentioned the synthetic lethality project with PKMYT1. So in a normal homologous recombination, the BRCA scaffolding protein is essential for DNA double-strand repair. And when this BRCA is inactivated, such as in cancer, this leads to deficiency of homologous recombination DNA repair, and then to progression of cancers, especially breast, ovarian, and pancreas cancers. And BRCA deficient tumors are treated with PARP inhibitors, which cause synthetic lethality and ultimately to cell death. But we also know that there is some emerging resistance as a major issue of those PARP inhibitors. So that's where Pol Theta is very interesting. It's a DNA repair enzyme, which is overexpressed in homologous recombination cancer cells, but not in normal tissues. And so as a monotherapy, we can use Pol Theta inhibitor to get some synthetic lethality and ultimately to cell death. And we can also use that as a combination therapy with a PARP inhibitor I just mentioned, plus Pol Theta inhibitor to also circumvent some resistance caused by PARP inhibitors and finally get also a synthetic lethal event. So for Pol Theta as a DNA repair enzyme, there is two druggable domains. One is the enzyme helicase, and the other one is DNA polymerase. And so we wanted to explore that target, as we know that there is already several inhibitors targeting both domains advanced in the clinic, such as the Artios 6043 you have on the right, with a pretty nice activity of 1 nanomolar, and which is being progressed in phase 2 by Artios. So for our drug discovery project, we were interested in trying to find new allosteric inhibitors of polymerase theta domain. So we used information from non-allosteric inhibitors such as the Artios 558, which has a more simpler chemotype compared to the previous one. And also there is the repair with the alkyne for an activity which is pretty similar. But you can also see on the 6 different examples that you have on the right that many companies have been working in this field and with pretty much the same pharmacophoric features, such as the one you have on the left. So the green part, which is a very lipophilic motif attached to a kind of a linker, and on the right, a methyl aniline that also goes into a lipophilic pocket. So based on that information from the Artios 558, we have built a kind of pharmacophore and also a contact score. So we are using those elements to drive our generation. And so we are basically focusing on key interactions within the binding site, such as the one depicted on the scheme. You have also the spheres of the pharmacophoric point that helps also for the docking pose to get a proper docking pose within this very lipophilic binding site. And so we have applied 3D GenAI fragment growing and linking approach associated with a robotic synthesis for the heat discovery part. And so on the scheme in the middle for the growing approach, we have used a similar group as the one depicted on the Artios. So the methyl aniline, for which we wanted to have something new as a linker and central scaffold. And we also decided to make a linking approach that will combine this fragment on the right plus a simplified version of the Artios having a methyl CF3 pyridine. And so having everything set with some 3D structure-based reward functions, I just mentioned pi stacking, hydrophobic edge bonding. We also added some phys-chem constraints, a very basic one, such as molecular weight or number of rings. And we finally ended up with 8 different scaffolds you have on the bottom right, such as indolene, indanone, pyridobenzoperoxazine. And there were also some interesting scaffolds that were also subsequently discovered by Dan Atlas. So we had this different scaffold in hand. And so basically, we picked almost 250 molecules, 86 were synthesized, and we finally came with 8 different chemotypes with activity ranging from 27 to higher than 50 micromolar. And as I just mentioned, compound 740 and 751 are the ones that have been subsequently reported by Dan Atlas and patented. So we had to drop off this very interesting chemotype. We also came with simpler ones, such as the hydroxyamide compound 737, and also some very simple compounds, such as the one in the middle, with activity around micromolar. And finally, we came up with the oxime 751, for which we were able to separate the 2 isomers. The E isomer being about 10-fold more potent compared to the Z, simply because with the OH of the oxime, we can recapitulate the glutamate interaction that was present in the Artios compound. So it was interesting to have this OH bond that do create a nice interaction and enhance the potency of the compound. And it's great also to see that this oxime, which is not super common in medicinal chemistry, popped up in the generation. It might be misleading to escape from this oxime because it's stable, and it has proved also, and we'll see in the results, that it's also metabolically stable. And eventually we had the biochemical activity confirmed on the Theta polymerase domain, as you can see with the curve on the left, with the Artios compounds also on the same graph. And ultimately, we profiled the oxime in a cellular proliferation assay, having the DLD isogenic cell line with a BRCA knockdown and the wild type. And so you can see in this proliferation assay after 6 days that the Artios 558 and especially our compounds 759 limited the proliferation with the confluency of cells being lower around 20%. And of course, in the wild type, there were no change at site with 30 micromolar of compound being tested. Then we decided to progress that oxime into a short heat expansion. We also wanted to balance the ADME properties of the compounds. So basically, we thought that having contacts with key polar residues, such as the serine on top or the arginine, might be fruitful to balance the ADME properties on top of increasing the potency. And it's actually the case. On the right-hand side on the table, you can see that compound 867, the amide, or compound 887, the carboxylic acid, turned to be super potent with nanomolar activity for the carboxylic acid. Also, the amide has this double interaction you can see on the minimization, docking minimization of the compound. And eventually, we progressed those two compounds in ADME profiling. So we had good hepatocyte clearance, human hepatocytes, even better than the Artios one. The solubility was perfectly fine. Keiko2 was not fantastic, not surprising considering the log-D, but still it's a very interesting compounds, and there is also the amide that turned to be much more permeable compared to the carboxylic acid. So finally, to conclude on that program, so we discovered through AI many scaffolds, especially 8 chemotypes, for which a novel oxime series was identified, especially through well-defined contact within the binding site. We had about a 2-log improvement in potency within 2 optimization cycles. And finally, the lead compound has a biochemical activity of about 10 nanomolar with a good ADME profile and a validation in a synthetic lethality mechanism. And overall, also, we made 86 molecules, and it turned that one lead series emerged after about 12 months to reach this E2 lead phase. And I will now present the PDL1 project. This was a collaboration with our Japanese partner, Kurayo. So PDL1 is an immune checkpoint protein, which is expressed in tumor cells. It is one of the most validated immuno-oncology targets, especially because it's overexpressed in multiple cancer types. And it has been correlated with a poor prognosis and very aggressive disease. And so the binding of PDL1 to PD1, especially in the context of monoclonal antibody, does suppress the T cell activity and allows the tumors to escape from the immune surveillance through different mechanism. But yeah, overall, it reduces the T cell proliferation and activation. It suppresses the cytokine production and promotes T cell exhaustion and apoptosis. And so on top of these monoclonal antibodies, for which we know that there is some liability, we wanted to explore more the small molecule opportunity to try to get some interesting chemical compounds that will not disrupt the interaction between PD1 and PDL1, but something with a differentiated mode of action, especially because the small molecule do induce the dimerization of PDL1, internalization, and eventually the degradation of the PDL1. So there is a strong interest for a small molecule with this type of mode of action, but actually no clinically compounds had advanced to after phase 2. Here there is some examples on the big players that were really involved in finding small molecule in this area. There's been more than 200 patents published. Most applications do keep this biphenyl core that you have on the different molecules. Compounds could be substituted on one or the two sides. So you have on the first picture compounds from BMS or Jubilance that do have only one side substituted. And on the right-hand side, compounds more sophisticated from Chemocentrics that was took over by Amgen, went to phase 2 with this both substitution on the two sides and also Insight that also went to phase 2. So basically, it's a super crowded area. But we also know that there is some SAR opportunity, there is some creativity to be made in this field, especially to modulate the DMPK, the potency, and finally to get IP. So we decided to combine efforts in terms of generative AI with structure-based information, scoring, and multi-parametric optimization, having a lot of information coming from the competition and from our partner. So we managed to build machine learning models for activity, such as the PPI biochem activity and SHIP1 cell-based assay in Jurkat cells, along with ADME parameters such as log-D, cytotox, microsomal stability in mouse, and Pampa. And we also used information from the competition to try to stay away of the known chemical space. And finally, the synthesis was made by our colleagues from Curayo, and you can appreciate the complexity of the molecule having this BRL system combined with two different branches on the right and on the left. So as a lead optimization phase, we use, as I mentioned, information from the competition plus the one from Curayo. So we have the starting compound having the BRL system and an amino-indane motif on the right. Compounds was potent at about 20 nanomolar. And so based on the binding interactions of this compound, we were able to build a kind of contact score to drive the generator to be able to keep those very key interactions, such as pi stacking in the center of the very lipophilic part, hydrophobic contact as well, and also very important H bonds, especially on the right-hand side, which are not completely solvent exposed, but they do contain some very important and nice H bonds. We do put also some physicochemical constraints, and eventually we managed to optimize this series with compounds having piperidine modifications, such as the one depicted on the right, with PPI activity ranging from 2 to 4 nanomolar. And we also identified a new scaffold, not as potent as the starting material, but still modification of the amino indane was doable, having a different heterocycle that could serve as a backup series. So the compound were optimized through different iterations with the machine learning prediction models that was trained on the data, enriched with the data that came in during the project. And so we were able at the end to get compound like the one on the right, 451, which was a racemate, so it could be even better. But still, the PPI biochem activity was about 5 nanomolar, the cell activity below 1 micromolar, with a low FG2 cytotox of 62 micromolar. Eventually, this compound and the series was not progressed due to a moderate bioavailability, and also compared to the inside compound, the cell-based assay was the activity of only 1 micromolar was very low. So we decided to switch to the modification on both sides of this BRL system. Again, we used information from the data sets to drive the multi-parametric optimization. So this time we had different type of modification. On top of the amino indane, there is also this aminopyrazole or the tetrahydro oxazolopyridine. Both compounds have activity around 200 nanomolar. But when we launched our prediction on the activity of these compounds combined with different motif and different exit vector and attachment, we ended up with compounds having, on one hand, amino indane and pyrazole with activity of around 7 nanomolar, and the other one, amino indane and THOP, for which the activity was as low as 2 nanomolar, which was pretty convincing to continue optimizing this series. So we further explored with different attachment to the piperidine, and we're able to find compound 551, for which the cell-based assay was below 100 nanomolar. Clearance was a bit high, of about 100 in mouse, and cytox was okay. And eventually, compound 467, having a pyrazole on the left and the amino indane, turned to be a very potent compound in both biochem and cell-based assay, and with a pretty good clearance in mouse and hepatotox. This compound has a bioavailability of about 15%. So just to conclude, so we have found and optimized a lead series with sub-nanomolar activity in the PPI inhibition cell-based assay was comparable to the competition with an acceptable PK. And we've really unlocked the chemical space using multi-parametric optimization, as those chemotype were not covered by the competition. And we also, within only 40 Iktos molecules out of 500 that were made through the project. So we accelerated very much the LO phase using this MPO approach. And so discovering all these chemotypes and having the 2 lead series took about 12 months to reach the LO stage. And we'll let Alexis discuss about our Iktos portfolio.
So here you have seen 4 examples of targets that are, of course, more or less well known, pretty well covered by many competition. They were to our end very much also a way to demonstrate the strengths of the different AI tools and technologies that we can develop in the context of drug discovery. But for our own and our current portfolio is based on targets that are completely different. But most of them are pretty challenging with sometimes nearly no molecule known. So we are more in the context of not just complete novelty over a crowded landscape, but more complete novelty full stop. We need to discover new molecules that does not exist in any way. So we have a program on a very challenging enzyme, MTHFD2, in immunoinflammation, a GPCR, for which we are targeting novel agonists, and a PPI program, SKIP2, CKS1, where there is no molecule known. And of course, as many companies, we are investigating the induced proximity and protein degradation area by the combination also of our technology, MTBench, which help us to have, let's say, a proprietary assay for induced proximity within the cell. And maybe also some acknowledgement of the many colleagues that did work with us.
So yeah, finally to conclude on this presentation, so we would like to thank all the colleagues we worked with. So we are very grateful to people from Oncodesign that were involved in the PIM project. Also, we thank people from Signature in the UK that were working on the Pol Theta program, and finally our Japanese colleagues that also made a fantastic job on PDL1 inhibitors on the synthetic and testing part. And also I would like to thank the Iktos team that was involved in the project, and thank you for your attention.